Hello there, Scribble Dog here. Welcome to my channel. Today I am going to talk about the tricarboxylic acid cycle. Tricarboxylic acid cycle, also known as citric acid cycle or Krebs cycle, is a key metabolic pathway that connects carbohydrates, fats, and protein metabolism. It is a central hub in cellular respiration, linking glycolysis and the electron transport chain. In eukaryotic cells, the citric acid cycle occurs in the matrix of the mitochondrion. Pyruvate, the end product of aerobic glycolysis, is a major source of acetyl-CoA. Other sources being beta oxidation of fatty acids and from branched-chain ketogenic amino acids such as valine, leucine, and isoleucine. Pyruvate must be transported from the cytosol across the inner mitochondria membrane by a specific pyruvate transporter called mitochondria pyruvate carrier MPC1 and MPC2. Pyruvate dehydrogenase complex then converts pyruvate to acetyl-CoA. The pyruvate dehydrogenase complex is a multimolecular aggregate of 5 enzymes, of which 3 enzymes, pyruvate decarboxylase or E1, dihydrolipoyl transacetylase or E2, and dihydrolipoyl dehydrogenase or E3, are involved in the conversion of pyruvate to acetyl-CoA. The remaining 2 enzymes, pyruvate dehydrogenase kinase and pyruvate dehydrogenase phosphatase, are regulatory enzymes. Pyruvate dehydrogenase complex has 5 coenzymes. These are thiamine pyrophosphate lipoic acid, coenzyme A, flavin adenine dinucleotide, and nicotinamide adenine dinucleotide. E1 requires thiamine pyrophosphate, TPP, E2 requires lipoic acid and CoA, and E3 requires FAD and NAD+. Regulation of pyruvate dehydrogenase complex. The 2 regulatory enzymes, pyruvate dehydrogenase kinase and pyruvate dehydrogenase phosphatase, that are part of the complex alternately activate and inactivate E1. Pyruvate dehydrogenase kinase, a cyclic AMP-independent kinase, phosphorylates E1 and thereby inhibits its activity. PDH phosphatase dephosphorylates and activates E1. PDH kinase is activated by ATP, acetyl-CoA, and NADH. Remember that activation of PDH kinase inhibits PDH-E1 activity, turning off the pyruvate dehydrogenase complex. Pyruvate is a potent inhibitor of PDH kinase. If pyruvate concentrations are elevated, E1 will be maximally active. Calcium is a strong activator of PDH phosphatase, stimulating E1 activity. This is particularly important in skeletal muscle, where release of calcium ions during contraction stimulates the pyruvate dehydrogenase complex and thereby energy production. Clinical correlation. Important clinical correlation related to the pyruvate dehydrogenase complex are congenital lactic acidosis, subacute necrotizing encephalomyelopathy or Lee syndrome, arsenic poisoning, thiamine deficiency, congenital lactic acidosis, deficiency in the E1 component of the pyruvate dehydrogenase complex, although rare, is the most common biochemical cause of congenital lactic acidosis. This results in an inability to convert pyruvate to acetyl-CoA, causing pyruvate to be shunted to lactic acid, a reaction catalyzed by lactate dehydrogenase. Symptoms are variable and include neurodegeneration, muscle spasticity, and early death in the neonatal onset form. Subacute necrotizing encephalomyelopathy or Lee syndrome, a rare progressive neurological disorder that is the result of defects in mitochondrial ATP production, primarily as a result of mutations in the PDH complex, the electron transport chain, or ATP synthase. Both nuclear and mitochondria DNA can be affected. Arsenic poison. In addition to inhibiting a pathway in glycolysis, arsenic poisoning is mainly due to inhibition of enzymes that require lipoic acid as a coenzyme, including E2 of the PDH complex, alpha-ketoglutarate dehydrogenase, and branched-chain alpha-keto acid dehydrogenase, A trivalent form of arsenic colored arsenate forms a stable complex with the thiol, SH, groups of lipoic acid, making that compound unavailable to serve as coenzyme. When arsenite binds to lipoic acid in the PDH complex, pyruvate and eventually lactate accumulates. Like pyruvate dehydrogenase deficiency, this affects the brain, which relies on the TCA cycle for most of its energy, causing neurologic disturbances and death. Thiamine deficiency. Deficiencies of thiamine, as seen in alcohol users or niacin, can cause serious central nervous system problems, as brain cells are unable to produce sufficient ATP via the TCA cycle if the PDH complex is inactive. Now let's talk about the reaction sequence of the TCA cycle. The reactions of the TCA cycle are carried out by 8 enzymes that completely oxidize acetate in the form of acetyl-CoA into 2 molecules each of carbon dioxide and water. The cycle begins with the synthesis of citrate from acetyl-CoA and oxaloacetate via condensation reaction catalyzed by citrate synthase. Citrate inhibits citrate synthase and phosphofructokinase, the rate-limiting enzyme of glycolysis, and it activates acetyl-CoA carboxylase, the rate-limiting enzyme of fatty acid synthesis. In the second reaction, citrate is isomerized to isocitrate by the enzyme aconitase, an iron-sulfur FeS protein. Clinical correlation. Aconitase is inhibited by fluoroacetate, a compound that is used as a rat poison. Fluoroacetate is converted to fluoroacetyl-CoA, which condenses with oxaloacetate to form fluorocitrate, a potent inhibitor of aconitase, resulting in citrate accumulation. Isocitrate then undergoes an irreversible oxidative decarboxylation, forming alpha-ketoglutarate, a reaction catalyzed by isocitrate dehydrogenase, yielding the first of 3 NADH molecules produced by the cycle, and the first release of carbon dioxide, CO2. This is one of the rate-limiting steps of the TCA cycle. Isocitrate dehydrogenase is allosterically activated by ADP and calcium 2+ ion, and is inhibited by ATP and NADH, whose levels are elevated when the cell has abundant energy stores. The next reaction is oxidative decarboxylation of alpha-ketoglutarate to succinyl-CoA is catalyzed by the alpha-ketoglutarate dehydrogenase complex. CO2 and NADH are also produced. Alpha-ketoglutarate dehydrogenase complex is similar to PDH complex. The coenzymes required are thiamine pyrophosphate, lipoic acid, FAD, NAD+, and CoA. Alpha-ketoglutarate dehydrogenase complex is inhibited by NADH and succinyl-CoA and activated by calcium 2+ ion. However, it is not regulated by phosphorylation-dephosphorylation reactions as in the pyruvate dehydrogenase complex. Of note, alpha-ketoglutarate is also produced by the oxidative deamination or transamination of the amino acid glutamate. The next reaction is cleavage of the high-energy thioester bond of succinyl-CoA by succinate thiokinase, also called succinyl-CoA synthetase, yields succinate. This reaction is coupled to phosphorylation of guanosine diphosphate, GDP, to guanosine triphosphate, GTP. GTP and ATP are energetically interconvertible by the nucleoside diphosphate kinase reaction, GTP plus ADP yielding GDP and ATP. This reaction is an example of substrate-level phosphorylation. Of note, succinyl-CoA is also produced from propionyl-CoA, derived from the metabolism of fatty acids with an odd number of carbon atoms, and from the metabolism of several amino acids. In the next reaction, succinate is then oxidized to fumarate by succinate dehydrogenase. Its coenzyme FAD is reduced to FADH2. Succinate dehydrogenase is the only enzyme of the TCA cycle that is embedded in the inner mitochondrial membrane, and it functions as complex 2 of the electron transport chain. Next, fumarate is hydrated to mal-8 in a freely reversible reaction catalyzed by fumarase, also called fumarate hydratase. Note, fumarate is also produced by the urea cycle, purine synthesis, and during catabolism of the amino acids phenylalanine and tyrosine. In the next reaction, malate is oxidized to oxaloacetate by malate dehydrogenase. This reaction produces NADH. The delta G of the reaction is positive, but the reaction is driven in the direction of oxaloacetate by the highly exogenic citrate synthase reaction. Of note, oxaloacetate is also produced by the transamination of the amino acid aspartic acid. At this point, it is worth mentioning that the TCA cycle does not involve net consumption or production of oxaloacetate or of any other intermediate. 2 carbon atoms enter the cycle as acetyl-CoA and leave as CO2. During one turn of the cycle, 3 NADH and 1 FADH2 are produced. Oxidation of 1 NADH by the electron transport chain leads to formation of approximately 3 ATP, whereas oxidation of FADH2 yields approximately 2 ATP. A total of 12 ATPs are produced, or 15 ATPs if you count the NADH from pyruvate dehydrogenase complex. Thanks for watching. If you enjoyed this video, don't forget to give it a thumbs up and hit the subscribe button for more content like this. You can also check out our other videos on the channel, and remember to turn on notifications so you never miss an upload. See you in the next video.